Good morning to all of you to today's biodiversity seminar. I'm very happy to welcome all of you and all the Zoom people, of course, too, and Irene Cordero especially, who will talk about soil biodiversity and its response to drought and land management today. Irene is working in my group as a tenure-track scientist since almost a year, started last May, a year ago. But before that, she did her PhD in Spain at the Complutense University in Madrid in collaboration with an agricultural science institute in Madrid as well. After that, she received the prestigious grant from Spain for her own postdoc fellowship to go to England, to the Manchester University. A lot of you may know Richard Barchett, that was her advisor in Manchester. And after those 2 years, she applied for another grant from the British Research Council, a discovery grant, and received that one for 3 years to continue her postdoctoral work in Manchester as well. And after that, she joined us last year. And very happy to hear about soils. The floor is yours.
Thank you. You wanted to introduce the... Okay. That's fine. Okay, thank you very much, Anita, for the introduction. And yeah, thank you to the Biodiversity Center for giving me the opportunity to present my research. I'm very happy to be here. And of course, as Anita said, I'm gonna talk about soil today. And I know some of you in the audience, perhaps here in the room or in the Zoom, you do work with soil, but perhaps also some of you, you don't work with soil, you work with other parts of the ecosystem and you haven't really given so much thought to the soil itself. But today I'm gonna invite you to join me and imagine yourself being a tiny, tiny little animal living in the soil and yeah, take this, this trip to the below ground and take a soil perspective, which is actually not a very common perspective. And in general, soils are really underappreciated. So many people just don't think of soils as an important part of the ecosystem. And others, even worse, they just think of soils as dirt, something you don't even want to touch. And that's terrible because soils are extremely important. They are ubiquitous, they are everywhere. They are, of course, the base of every single terrestrial ecosystem, and they are steaming with life. In a single spoonful of soil, you can find more individuals than people living in this planet, which is quite a striking number. And other numbers that we can see here, for example, in the same spoonful of soil, you can have 1 billion bacterial cells, you can have several thousands of different taxa, and even up to 200 meters of fungal hyphae. So these are very astonishing numbers. But this not only these numbers are impressive, soil biodiversity is also very, very beautiful. I hope you'll agree with me. And the biodiversity spans the whole tree of life. It can go from the tiny microscopic bacterial cells to the fungi that can be also microscopic cells, but they can also create these massive networks of hyphae that spread below ground, communicating plants and trees all through the forest. You can have many of those nematodes here on the right side. They do many different functions in all the trophic levels in the soil food webs, from fungivores to predators. You can also find some of these weird creatures on the left, the tardigrades. They are tiny animals that can withstand impressive harsh environments. And you can also see slightly bigger things, also these beautiful springtails and mites in the middle line. They also can be detritivores, they can be predators, and you can go to bigger things, millipedes, centipedes, earthworms, or even some small mammals. So you see the biodiversity in the soil spans a really wide range. And they say that 40% of the terrestrial biodiversity is in connection with the soil, at least in one part of their life cycle, if not their whole life cycle. So 40% is a really high number. And that is why we should really consider the soil when we are thinking on biodiversity conservation strategies. And this high biodiversity is also doing a lot of functions in the soil. So the soil and all the inhabitants of the soil together, they provide what we call the ecosystem services. So there are different things, such as organic matter decomposition and nutrient cycling. So moving the carbon, nitrogen, phosphorus, other micronutrients in the soil. They, of course, can take the carbon, put it in the soil, so the so-called carbon sequestration. They provide pure and clean water, and they support plant growth. So they do a lot of things. So that's why the soils are vital and for the ecosystems and for humanity. And they really play a key role in our food security, and they can help us a lot with the climate change mitigation strategies, and of course, as I said, the biodiversity conservation. But to be able to provide all these services, the soil and their microbial communities need to be, their organic biotic communities, sorry, they need to be healthy. However, we all know that they are increasingly challenged by human-induced perturbations, such as climate change or land use intensification. So climate change, we all know what is climate change in this room. The planet is warming, and with that, we are also experiencing more extreme weather events. In the last IPCC report a couple of years ago, they published this map where they simulated what is going to be the moisture content in the soil, predicted moisture content in the future years. And we can see that some areas of the globe are getting wetter, but some other areas are getting considerably drier. And that includes almost all Europe, where we are. And droughts are not only getting more intense, they are also becoming more frequent. In the same report, they also published this figure where we can see the frequency of these extreme drought events. So one event that in the past used to happen only once every 10 years is predicted to happen up to 4 times every 10 years. So when we are thinking about drought and the effects of drought in the ecosystems, we need to think of the intensity and the frequency in combination. The second big factor that is affecting the biodiversity is the land management intensification. So 50% of our habitable land is used for agriculture, as you can see here in the graph, and most of it are pastures for livestock, for grazing, for hay production. So it is a very big amount of land. And of course, to be able to feed this ever-growing population, what managers and farmers have been doing is just to intensify the production. So put more cows, put more sheep in the same land, or add more fertilizer, add some more pesticides. And we all know that this intensification has negative effects in other variables, other things in the ecosystem. So for example, we can see here, as we increase the land use intensity, the biodiversity of plants significantly decreased. And this happens not only above ground, it also happens below ground. So I focus all my research in these 2 main things affecting the biodiversity. So the drought and the land use intensification. So today I wanted to summarize my research in these 3 parts with increasing complexity. So first, I'm going to talk to you about experiments where I only had soil, so bare soils in pots, and really focusing on the drought effects. Then we move into a plant in the pot. So it's still a simplified system, but we have the plant on it, and we also focus, I will talk you about some other factors that can affect the biodiversity, so plant species identity or land management. And then in the last part of the presentation, I will talk you about complex mixed plant communities in bigger pots and more factors affecting it. But step by step, let's start with part 1. So we all know, and it's a lot been written in the literature, that soil biotic communities are sensitive to drought. Some groups of organisms are more sensitive than others, and so the balance can change, but in general, as a community, they are really affected by drought. And one thing that can happen is that the community can change to what is called an alternative stable state. So this theory of the alternative stable states is better understood if we think of the ecosystem as a ball that is moving within those 2 valleys. So when we have perfect non-drought conditions, The pointer is not working, as always. There we go. Okay, so yeah, we have this beautiful ecosystem with no problem with drought. We are in the reference regime, this white part. Well, beautiful, it's not very diverse, but it's not droughted. So we have the ball in this valley. And then if there is a perturbation, the ball may move a little bit up or down. But if the perturbation is not strong enough, then the ball will fall again and will stay stable in this regime. But if we increase the perturbation, the ball will start to move upwards, and then it may reach a tipping point. And if we keep pushing the system, we cross a threshold in the perturbation, and the ball will fall into the other basin. So in the, in the alternative regime here. And once the system is there, it will be very, very difficult to go back, even if we stop the perturbation. So that's why it's called alternative stable states. And this theoretical framework has been demonstrated for different ecosystems, mostly the aquatic ones, so in lakes and in coral reefs. Also some plant communities with these grasslands, dropland with influenced by fire regimes. But it has proved quite challenging for soil communities, because one of the reasons is that the soil communities are extremely complex. And we won't have very beautiful two valleys. We will have something like this mountain range full of many, many valleys and many peaks. And these complex connections and networks of species that makes all this understanding of what is going on quite challenging. But still, we should be able to see a difference of the original community compared to the community after the perturbation, and see if there is a coming back after the recovery or not. So I decided to design an experiment to test this. And I took these soils and put them in these little pots with no plants, as I said, and imposed them a matrix of drought, intensity, and frequency with 3 levels of drought. So a mild drought, an intermediate drought, or a very intense one, and 3 different frequencies. So just 1 event, 2 events, or 3 events, one after the other. The drought period lasted like around 2 months. And then I started taking samplings. So just after the rewetting, and then 1 month, 3 months, and 6 months after the end of the drought to follow up this recovery. And what I found out is that there was a permanent shift in the microbial community. So this data is the biodiversity after 6 months. And we can still see that the bacterial diversity has decreased and the fungal diversity has increased. And is there, it's not going back. And when we look at the community structure, so we see here with the different colors is the different drought treatments. So we can see all the reds on the right-hand side of the graph, which is the control. And then as we move through the gradient of drought with more intensity and more frequency, we move to the left side with finishing these pink purple dots that are all on the left side. And with the different shapes here, we can see the time points. So the circles are just after the end of the drought, but the triangles and the squares are after the drought, and then we can see they are not coming back. Basically, every time point is in the same area. So we can understand this as a permanent shift in the community. And this shift in the taxonomy was translated in a shift in the functionality. So here I show you some data for some soil enzymes, which are enzymes in charge of breaking down the organic matter in the soil, such as the chitin, the lignin, the cellulose, some different pools of inorganic nutrients and organic nutrients in the soil, and carbon, nitrogen, and phosphorus in the microbial biomass. And the colors means when there is a yellow, there is no change compared to the undroughted control. The red means there is a decrease, and the green is an increase. So as we move to the right side of the graph, we can see that the yellows start to turn red, except for 2 variables, the ammonium and the DOC, which they picked. So after the drought, there was a very big peak on ammonium and DOC. This is a quite well-known phenomenon that usually when you rewet dry soil, the dead cells that are there release the contents of their inside. So you get this nutrient flush. But then, so this is what happened just after the drought. But after 6 months, the picture was really similar. The only thing that changed was this peak in the nutrient flush that was mostly gone. But even though there was loads of nutrients available, it was not translated into more microbial biomass. The microbial biomass was still significantly reduced. And also the functionality. So the functionality of the system was not recovered. Even more, we saw some type of adaptation of the community to the new conditions. So to study this, we decided to take those soils and impose them a new full dry and rewetting. So we completely dry every single soil and rewet it and follow the bacterial and the fungal growth through the incorporation of radioisotopes. And what we saw is that the soil that has this history of previous drought, the bacteria community tend to grow more. So here on the left, so we see these increases. So they accumulated in general, it's a bit of variability, but they accumulated more biomass when they have this history of a previous drought. And in the middle graph, this lag period means the time it takes since you add the water until the community start to grow. So the bacteria decrease this period. So they started to grow faster. So basically the bacterial community somehow restructured and adapted to a fast recolonization after drought. On the other side, the fungi show the opposite. They grew less after the intense drought. Perhaps it's a strategy to kind of preserve resources for the upcoming harsh conditions. We don't know exactly, but it's completely opposite to the bacteria. So with all this, we can say that after this very intense drought, we're probably having a new microbial state. And we can visualize it here. This is all the functions again, the enzymes, the nutrients, and the microbial biomass with the same colors for the drought treatments and different shapes for the time points. So if we go back to this image of the ball inside this valley, the black circle is where all the red dots are. So that is our reference regime. So all the time our control pots were in there. And then if we apply an intermediate drought, we see some blue dots going out of the circle. But then when we look at the crosses, which are the 6 months, all the crosses are back within the original circle. So we can think of the system going a little bit out, but bouncing back. However, with the intense drought, we see that the pink dots go out in the same direction as the blues, but much farther away. And then when we look at the crosses, they are just in a different area. So we can interpret this as our new alternative regime. So this is very interesting. It's a proof of concept experiment, let's say, with these bare soils. But we know that the microbes do not live in isolation. They interact with many other organisms, and especially with plants. Plants have a massive effect on the microbial communities through their leaf and root litter, and through the root exudates. They can really change and shape the microbial community in the soils. So this leads us to the second part of the presentation, where I did some experiments with the plant in the system. And I also want to point out to two important factors that I studied: the plant species identity and the land management. So in this figure, we have, again, similar colors for the different drought treatments, again, with a gradient of intensity and frequencies. And we can see, again, this is bacterial data, so 16S sequence data, that the bacterial community is affected by the drought. So we have all the red dots on the left side moving towards the right side through the gradient of drought. But very interestingly, I don't know if you can really see, but the two grasses have different shapes. One is a circle, the other one is the triangle. And so I planted two different grass species here, Anthoxanthum odoratum and Holcus lanatus. The difference between those two grasses is their growth strategy. So Holcus lanatus is a fast grower grass. So they differentiate in many different root traits and leaf traits. But it is said that the fast-growing plants in general are more sensitive to drought. And here we can observe that the pink triangle here, the blue triangle here, so they are farther away than the circles. So this means that it's not only the plant that is more affected by the drought, but the microbial community in the soil that this plant creates is also more affected by the drought. So this is really interesting. And it's not only the plant, it's also the management. So here again, we see the same gradient of the drought, and the intensive is the triangle. So for this experiment, I collected soil in different farms. They have different levels of management. So it's long-term management of the soil. I divided them in these two categories. So one is more intensive, so there's a higher stocking rate, they add some fertilizers and they add some seeds. And the extensive, there was no fertilizer, no seed addition, and a little bit of grazing, but not a lot. So that's how I differentiate them. And we can see the triangles are the intensive, that again, the triangles are farther away. So this also brings a very important message, perhaps, to share with farmers, that if you treat your land very intensively, then the system, or at least the bacterial communities, are going to be less stable under drought, under future climatic conditions, as predicted. And this is not only the taxonomy. Again, when we look at the functionality, this is again soil enzymes, microbial biomass, etc. Similar pictures. So there is a drought gradient, and the effect of the plant is even bigger. So we can see the triangles, the two triangles here, completely apart, and the Anthoxanthum, the slow-growing plant, doesn't have that strong effect. So again, the fast-growing plant, much more affected. And the same for the management here. I decided to color the dots by the management, so you can clearly see how the soils coming from different management farm differentiate themselves very clearly regarding their functionality. So we have seen that the drought, the plant species that is growing in the soil, and the management are all affecting our microbial communities in the soil. And my next step was to ask, okay, what are the consequences of this? So the plant is affecting my soil, but is my soil affecting the plant back? So is the soil able to change somehow the performance of my plants? And to do that, I did some whole soil inoculation. So basically you take the whole community of the soil, you take a soil, in like a 5% of your soil, and you put it in a new pot with a sterile soil, and you plant a new plant in there, and you see how the plant responds in performance, how they grow, how they react to a new drought. When you talk about inoculation, many people think of individual strains. So I've done this in the past also quite a lot. So you select a specific bacterial strain that you have seen that is drought tolerant or whatever, stress tolerant, and you inoculate it into a plant. And that is it's a lot of literature on that because of the importance for the economy, for the crops. For example, I'm just going to show you one figure from a previous paper where I selected 3 bacterial strains that I isolated from a dry environment, and I inoculate them into a plant. And then here I'm showing the relative leaf water content. So this shows how much is the plant basically suffering from the drought. So the uninoculated control, it declined around 20% their water content when you put the plant into drought. When you have a chemically fertilized plant with NPK fertilizer, the decline is much stronger, 40–50% decline. But when you inoculate your plant with these biofertilization strains, they really don't suffer much from the drought, or not at all in some cases. So these bacterial strains are really protecting the plant from the drought somehow. There are many different mechanisms behind, but that is happening. But okay, this is one bacteria. What happens when you have the whole soil with loads of bacteria, loads of fungi, nematodes, everything? What I found out in my experiments is that actually the whole community is doing a negative effect on the plant performance. So the plants grew less when they were inoculated. And quite interestingly, what I'm showing in this figure is that this reduction of growth was related with land management. So when we had a highly intensively managed site, here, this is the below ground biomass, so the root biomass was much lower, well, significantly lower, let's say, especially when the plant was under drought, than when the soil was coming from an extensively managed site. If we look at one of the variables that represent plant performance, so in this case, chlorophyll content, we can see here on the right side, the sterile plants are really doing much better than any of the other inoculated plants when they are under drought, not under control conditions. And it really didn't matter what was the origin of the inoculum. So my hypothesis initially was that if I have a soil that it has a history of a very frequent drought or a very intense drought, we have seen that the microbial community has changed. It has different bacteria, it has different fungi. Probably those bacteria and fungi are capable to keep themselves in better shape under drought, but they are not really affecting the plant. So that was a bit disappointing, but that's science. So yeah, the negative effect of the inoculum in general that we see is probably what is commonly called a negative plant soil feedback. So this means when there is a plant, especially for grasses, is quite well described, it's negative. So you have a grass growing in the soil and it tends to accumulate pathogens that are very specific for that plant species. So if you take that soil and you plant it again with a new plant of the same species, the pathogens are going to affect the plant and make it reduce the performance. So that's probably what we're seeing here, a negative plant soil feedback. Okay, so moving on to the last part of the presentation, I decided to work with these more complex grass communities, try to get a bit closer to the reality. And to make it even more interesting, instead of just taking the soil, sieving it, and planting it with the species I decided to just took intact soil cores. That's what we call. So it's like a ring, a bit of a pipe, a plastic ring that we hammer into the soil, and we just take the whole thing out. And then you get basically the intact or as intact as possible soil structure with all the roots in it and all the plants that naturally grow on that soil. And then we applied many different treatments on it because in the experiment I'm going to show you, what we are interested really is in to see what is the interactive effects. So you can have, for example, this is a theoretical graph. So imagine this is a response variable of any kind. It can be soil respiration, it can be pathogen accumulation, it can be anything. And our control soils respond like this. And then we have two different factors. It could be warming and drought, for example, but you apply them individually and you observe an increase in this particular response variable, one more than the other. Okay. What happens when you put those two factors together at the same time in the same soil? They can respond with an additive effect. So basically, what is the effect of one factor plus the effect of the second one? However, these other options, you can have an antagonist effect. So somehow the two factors interact and making the response not so strong. It can be a dominative effect. So just the factor that has the strongest response is the one determining what is going to be the result in the variable. Or it could be a synergistic effect. And suddenly you have a massive increase or decrease of a certain variable that cannot be explained by the individual effects. And this is sometimes called the ecosystem surprise. So you put things together and ta-da. But this synergistic effect has been shown, it happens in the nature, and it happens quite often. And it's even more frequent when you start to add more and more factors, one on top of the other. So that is one thing that we wanted to understand. And in this experiment, that was my first experiment here at VSL, and we did it this summer in the greenhouses. We applied those 10 different perturbations. So we add some nutrients, so added nitrogen and phosphorus independently. We had 2 treatments related with grazing, so a defoliation treatment and a trampling treatment. We have some pesticide additions, so insecticide, fungicide, herbicide, and antibiotic addition. That's also important because if you have some cattle that's been treated with antibiotics, you put it out into your pasture, they defecate, and then the antibiotic is in the soil. And so all of those are related with land management in grasslands, and the last 2 related with climate change. So we had a heat wave and we had a drought treatment. And I'm showing you 3 response variables. So the amount of nitrate in the soil, in the middle, the ecosystem net assimilation, so how much CO2 was taken from the whole system, so all the photosynthesis minus the respiration. And finally, the decomposition rate through the TVAC index. So the individual perturbation, that's how they affected these 3 response variables. For the nitrate, there was not a big effect, just 2 of them increased. Luckily, the nitrogen addition increased the nitrogen content. The other 2, we have some perturbations that increase this value, others decrease the value, so a bit of variability. But when we started to add them together, so what we did, we combined them in like 2 of them, 5 of them, 8 of them, or all of them, a bit random within each group. And we saw a different picture. And in the nitrates, we can clearly see the synergistic effects I was talking about. It's a very, very big increase in the amount of nitrogen in the soil. With the net assimilation and the decomposition, interestingly, there were variables going a bit higher, a bit lower, but when you combine them, they are all going lower. And this is probably a dominative effect of the drought, which seems to be the factor that is most affecting both of them in this case. But this is a proof, basically, that it is very important to understand what is happening when you add different factors. This is not only one, it's important, but this is also a bit real life. In real life, you will rarely find one single perturbation happening. If you have a natural grassland, you may have a bit of warming, then that leads to a bit of drought, you may have some nitrogen deposition. But then if you have a pasture, it's even worse, you have all these management things on top of each other. So it is important to understand these combinations and interaction of treatments. Okay, so far at this point of the presentation, perhaps you're starting to think, hey, in the first slide you show us some beautiful collembolas, some beautiful mites, where are they? And it is true. So far, my research has really focused on fungi and bacteria, which are the base of the soil food web. And they are extremely important, but they are not the only actors. And lately, especially since I joined the VSL in Anita's group, we are trying to look a bit more into the whole soil food web. Well, probably not the whole, but expand a little bit. In this experiment I did this summer, we already have some data on nematodes and some data on some microarthropods that will be analyzed at some point. But it is important to take this holistic point of view and try to understand the food web, because they are all together the ones that provide the ecosystem services. It's not only the microbes. We have seen on my own research and the research that other people have done that the drought and the different management practices can really affect the communities. They can really affect the provisioning of these services. So ultimately, like my goal with my research in general is try to find the balance on what is the best management level that we can keep, that will preserve the biodiversity, perhaps not enhance it, although sometimes it can enhance it, but at least preserve it, and that they will still be able to provide all these ecosystem services. And more importantly, that will keep the system stable under drought, keep it resistant, keep it resilient, and that is the real challenge. And with this, I think I was a bit quick, sorry. But yeah, I just wanted to finish thanking everyone that helped me along the way. There's a big list of co-authors, but many other people helped me with my previous home and the Soil Ecology Lab in Manchester, my new home, the Trier Pflanzen Interaktionen, I think I pronounced it right, at BSL. And yeah, of course, the funding bodies that funded my research, and all of you for your attention. Thank you.
Thank you very much, Irene, for an awesome talk about soils. I'm sure there's lots of questions, so please.
Thank you, Irene, for this really nice talk, and especially for taking the soil perspective. I'd like to hear that. I have a question to your first part, where you had this differently intense droughts, and you found that especially the intense drought brings your system into a new equilibrium. Can you distinguish from your experiments whether this is the drought effect or is it the rewetting effect? Or have you tried maybe with different intensities of rewetting, because then you might get this out?
That's a very good question, and I don't know the answer. No, my rewetting was the same for all of them. For me, I basically consider the perturbation, the drought, and the rewetting, like the whole thing together, because they are together. Because yeah, more intense drought, then you can see things happening, but it's also a more intense rewetting, of course. So yeah, no, I don't know, but yeah, it's really is one of the important things. When you start to get water again in the system, cells start to break, and it's a mess. But yeah, sorry.
Well, thank you very much. I was wondering, instead of looking at the beta diversity in the first and second experiment, did you try to look at the composition to see if you have specific species or OTUs or anything that is associated with your treatments?
Yes, there were specific changes in the composition. I think, now I'm just trying to remember, but with the control communities were basically co-dominated by proteobacteria and acidobacteria. But then after the drought, I think it was massively dominated by proteobacteria. So it was definitely some specific taxa that changed. But I mean, there were loads of changes. So it was difficult to have a deep understanding of all the things that were happening. But yeah, definitely there were specific groups. And for the fungi, there was specifically, I can't remember the name, but there were two families of fungi that they are in charge of organic matter decomposition. And those two families were the dominant ones, basically, in the control soils. And when the drought happened, they didn't disappear, but they reduced a lot. So it was kind of also a change, in the proportions of what you have. Yes, I also look into functional guilds and functional traits in the fungi, not in the bacteria. But yeah, it was the detritivores, the decomposers, not the detritivores, the decomposers, the one that saprotrophs, that was the word, the saprotrophs that really declined in the fungal. But I was a tiny increase. So in comparison, if you lose a guild, you increase the other one. So it was a tiny increase in the pathogens and the symbiotrophs. But I think it was just driven by the fact that those two big families of fungi really decreased.
We have one question on Zoom, then we continue in the room.
Yes, Tess Hayton asked, from your research, what do you predict the effect of drought and alternative stable states will be on soil carbon respiration?
On soil carbon respiration. So I think there was, of course, when you start with the rewetting, there is a big peak of soil respiration. But I think in general, all the functions were really declined. So the enzymes declined and all the decomposition declined. So I will say after the big rewetting pulse, I will think that the respiration will also decline. So yeah, that means that more soil is more carbon is staying in the soil. Perhaps that's perhaps good news. We don't really know, but no, I didn't really measure the soil respiration.
Thanks for this very interesting talk. I was wondering, have you ever looked to, or do you intend to look to the mechanical perturbations like plowing or soil compaction combined with droughts?
Okay. Yeah, I think it's something very important because yeah, the soil structure is super important to keep the water. So if you, for example, do plowing, you disrupt all these aggregates. So usually you are able to store less water in the soil, and the opposite when you compact. So we did have this trampling effect that compacted the soil. But yeah, I think there is a really big interaction there. It's something I will look more into the future, but it really changes. Yes, thank you.
Sports.
Thanks. I have a question regarding the different plant traits. So you planted species with like rather fast and rather slow traits. And you said that the fast-growing species are more affected by drought. They can also recover better. Did you look at that also? Maybe it was too quick and I didn't get it.
Yes,
Thanks.
I also had recovery time points. I haven't shown any data from that. So yeah, you are completely right. The fast-growing plants tend to suffer much, but they also recover. But in my experiment, they did recover, but not completely. They were still, if you compare with the slow-growing plant, they were more affected still. So yeah, I guess it really depends on the species that you choose. But yeah, thank you. Christian.
This is maybe not a question, and maybe it's too trivial for you, but I noticed when you apply your treatments, your drought treatment, for example, that not only the patterns change, the direction, the intensity, or concentrations, but also the error range often increases. And for me, this shows a bit that maybe these events, these extreme events, are a bit stochastic, they are a bit random, and what you get afterwards is really hard to predict. And I think that's also something we can learn from these experiments, that it's not only that things change, but also we don't know what maybe will happen. And that's quite interesting. Do you see that repeatedly?
Yes, and I completely agree with you. That's something that drives you crazy when you try to do stats. You have this difference in the variance. And it is true, yeah, the controls soils usually tend to have lower variability within treatment, but this gets bigger as you impose whatever treatment, honestly. And yeah, it's something I've seen repeatedly. And there are specific statistical tests that can look into that, because as you said, yeah, it's this stochastic effect. So suddenly in one of the pots, a whole family of whatever bacteria die, but not in the other one. And when they start to re-wet, then yeah, things don't look the same, but yeah, so you really increase the variability within treatments with the treatments. Yeah, that's a very good point. Thank you.
We have one question here from Yong Sang, who says, "Hi Irene, thanks for your amazing talk. I'm very curious about why the bacterial diversity decreased while the fungi diversity increased at intensively drought events."
Okay, thank you. Yes, so I don't know exactly why the bacterial diversity declined. I guess when it's especially the groups that were not so abundant in the control pots that died. So then you lose biodiversity, basically. Interestingly, what I showed is Shannon diversity. So for the fungi, it's not really that there were more species, they were more even. So we had these two fungal families I was talking before that were super abundant in the control. As they reduced their numbers, the evenness of the community increased. So that's why the fungal diversity seemed to increase. But of course, this is a pot experiment. You don't have newcomers. So we didn't really have new fungal species. But it's just, yeah, the evenness of the community. Good question.
So you did this cool drought experiment, and and you obviously had to choose a point at which to say, I'm done with the experiment. And I wonder if that point was motivated somehow by natural variation you see out in nature, but maybe 6 months was too long because it's already here, maybe gotten cold. And then if you were to imagine that during this drought, there was a drought and then it got cold. I was just wondering how like the temperature and the timing of the drought and all that interact. And if you would actually mirror sort of what we observe, say in Switzerland, would you get the same result or would you somehow get something different?
Yeah, I think so. It was kind of, in a way, it was motivated by the length of the growing season in England, in northern England. I mean, it also gets cold at some point, but it can be more or less 6 months. But it's also, we wanted to push it a bit, like let it keep going, because of course this is an incubation experiment. We have the perfect temperature throughout the time. So we wanted to see, okay, if we let it recover even longer, let's see if it recovers. But we didn't see any recovery 1 month, 3 months. And then we measured the 6 months and said, okay, no recovery, we finished with this. But yeah, no, it kind of resembles the length of the growing season. Here it is shorter, but yeah, I feel it will be the same. It will be the same.
More questions? I may have one for you. You
Yes.
showed that these bacteria get kicked into an alternative state or the communities. Would this, I mean, they don't go back, but would this be a bad thing or a good thing? Maybe that's more of a philosophical question, but could we have that new alternative state? Could that potentially serve the ecosystem better in the long run? Because these are species that may be drought adapted and would help the grassland to continue, or is change necessarily bad, maybe?
Exactly. I think it's a bit philosophical. But no, I think I agree with you. It can be something positive. And it's also kind of perhaps related with the question we had on Zoom about the soil respiration. Well, I mean, it's a different community. We're losing biodiversity, especially bacterial diversity. So that is not good. But the fact that the organic matter is decomposing a bit slower, perhaps is a good thing. So yeah, but of course, in the real life, we'll have plants there that will kind of help building up new organic matter. So we don't really know how it will really work. But yeah, perhaps is the way forward. Get some really drought-adapted bacterial communities, and they will kind of mitigate a bit the effects of the climate change. We'll see.
More questions?
One more here.
Okay.
Yeah. Yes, Natalie Oran says, thank you for the brilliant presentation, Irene. Very impressive. In your second experiment, including Anthocyanthum and Holgus, I'm wondering, what do you think is underlying these plant effects? And perhaps I miss it. How did plant species affect functioning?
Affect what?
Functioning. Functioning.
Functions. Yeah. Okay. Thank you, Natalie. So yeah, I think the thing is the Holgus is growing very quickly. So it produces a lot of leaf biomass, a lot of above ground biomass when it's still not very drought. So as soon as, so it's probably increasing the water demand for the plant itself. So you have a plant that is growing very big, very quickly, then it starts to evapotranspirate, and yeah, it increases the water demand. So I was watering the plants to a specific level, of course, so I kept the level similar for both plant species, but at some point between a watering day and the next watering day, it drops. I think really the fast growing species, that is what it's doing, is just using so much water, then at some point it breathes more water than the other one. So that's why it really declines. And yeah, the functionality, I think Anthocyanthum didn't really, so it changed from the control to all of the 4 drought treatments, but it didn't really affect that much. It was in the Holgus one that really had a very big difference between the control and the intense drought. The intermediate drought in the function didn't really affect that much. Good.
More questions?
Thank you, Irene, for your talk. Well, at the end of your talk, I think you said that you are trying to know the best management according to drug issues or fertilization. So the thing is, are you considering how to bring your results to the real life or the real situation? I don't know if you are considering to get a contact with some organization or public organization, or I don't know. Yes.
Yeah, I mean, of course, I think it will be amazing if these messages get to the landowners, mostly the farmers and the people in charge, or stakeholders in general. Yeah, in my future research, I plan to keep going in this direction, and my idea is to get in contact with lots of farmers here in Switzerland and start to see how are the soils in their lands, and how little changes, because you cannot really implement, okay, you have to change completely what you're doing. They're doing wrong. No, this is not the way, of course, and they are not doing wrong. They are doing what they consider is the best. But if small changes can perhaps, so if small changes can really change the stability of the system to the drought, that is my point. So perhaps when I look at all this combination of things and I say, okay, if I remove one of the things out of the system, so don't use whatever pesticide, and then the things look much better, perhaps. Okay, that's great. That's one way forward. Or if I do, so I can also look at the land management with some like continuous index that can help you look at this threshold. And if you see, okay, well, after you keep increasing the number of sheep in your land or the amount of fertilizer, so this is the threshold that after that, the system is not stable anymore. So try to keep it under this threshold. But yeah, of course, ideally, I would love to keep in contact with lots of farmers and share these ideas and see what they think. Yeah, that's the way in the future. Thanks.
Yeah, I was also wondering, I'm very surprised about the fact that it's your plant that is growing better on sterilized soil, right?
The plant is going, sorry?
You use sterilized
soil, yes.
And it's growing better?
Yeah.
So very surprised, but
yes.
I'm wondering, did you look at the nutrient? Well, how did you sterilize your soil? And after sterilize it, did you look at the nutrient content and if it was the same, if the sterilization did not affect?
Yes. So of course, yeah, sterilization affects a lot the nutrients in the soil. So we autoclave the soil. But after autoclaving, we flush it with lots of sterile water to try to reduce the nutrient content. And I measure the nutrients, so nitrogen and phosphorus mostly, just before, after the autoclaving, and then after the flushing with the water. And we did have a massive peak, but with the flushing, we lose quite a lot. Not everything, but quite a lot. But the thing is, I treated all my pots the same. And the ones that have the soil inoculum, it was just 5% of the volume. So 95% of the volume was this autoclave high nutrient soil, and it's just 5%. And I don't think that 5% could make such a big difference with the plant performance. I think it was really the pathogens in the soil. But yeah, it's a shame.
Thank you very much, Irene. And thank you all for your question, for being here, also the ones in the Zoom.